channel. Today we are going to discuss about the second part of meiosis, that is meiosis. What have happened after meiosis 1, that is prophase 1, metaphase 1, anaphase 1, telophase 1. After that we have discussed in the previous video. Today we are going to discuss about the second part of the video, that is meiosis. That is meiosis 2. As that of the meiosis 1, the meiosis 2 is also divided into 4 stages. First is prophase 2. Second is metaphase 2. Third is anaphase 2. And last is the telophase 2. If you remember, in the previous video I have told you, when you are mentioning about the meiosis 1, then write prophase 1, telophase 1 like that. In the meiosis 2 we are mentioning this. Why? Because the events that are occurring in both meiosis 1 and meiosis 2 may be somewhat similar, but certain differences are also there. So why I am discussing, if you want to know what is the difference, then I recommend you to watch the first part of this video, that is the meiosis 1. Before that also I have made one video, that is mitosis, about mitosis, and also in the beginning and the introduction part we have discussed about what is interphase. So now what will happen after the prophase 1 is, after this meiosis 1 is over, this prophase 2 will begin. What will happen in the prophase 2? The prophase 2, the chromosome, as you remember, during the telophase 1, the chromosomes were means returned back to the uncondensed form, and they were uncondensed form after the telophase 1. Okay, after the telophase 1 they have returned back to the uncondensed form. So then what will happen? Again the same process will happen in the prophase 2. That is the disappearance of the nuclear membrane, and the chromosome will start becoming condensed and the spindle fiber will start forming. As that it was happening in the prophase 1. But by this, if you remember, by the end of telophase 1, I have discussed that already 2 daughter cells were already formed. Already 2 daughter cells were formed. Okay, 2 daughter cells were formed. Now this prophase 2 will begin from these 2 daughter cells only, not from the 1. Remember that. From meiosis 1, single cell. That after the end of telophase 1, 2 daughter cells was formed. Now this prophase 2 will begin from these 2 daughter cells. Okay, chromosomes becomes shortened. Shortened means condensed. Okay, shortened and thicker. The thing which gets shortened and thicker, instead of that you can also use this word condensed, means it will become closer and compact structure. Okay. Second, what will happen? The nucleus and the nuclear membrane. Okay, the nucleus and nuclear membrane will start disappearing as it was happening in the prophase 1. But like that, what is the difference between prophase 1 of the meiosis and prophase 2 of the meiosis 2? If you remember, the prophase 1 was further divided into different stages: leptotin, zygotin, pachytin, dikinesis. Okay, but here it is not divided into any substages. Directly thus things will occur. Okay, so chromosome becomes shortened and thicker, nucleus or nuclear membrane start disappearing, and spindle fiber will start forming. Okay, will start forming. As I have told you, why the spindle fiber is required? Because after this stage, the second stage is the metaphase stage. During the metaphase, these chromosomes will arrange themselves in the equatorial plane attached to this spindle fiber, so that during the anaphase, when the chromosomes will be pulled apart with the help of cells, this spindle fiber only will contract and pull apart these two chromosomes. Okay. And now this is all about the prophase 2. I have told you the chromosomes will start arranging themselves in the equator. Okay, if this is a cell, then as I have told you, they are divided into two daughter cells. So these will be starts appearing, and what will happen? This is the chromosome. Okay, they will start appearing in the equatorial plane. Equatorial plane in the symmetric manner, still attached to the spindle fiber with the help of the centromere. From the centromere, they will be attached to the spindle fiber. Okay. Now you know why I am drawing these two daughter cells, because by the end of telophase 1, we have already came with these two daughter cells. So they are attached to the spindle fiber, and this is the stage of the metaphase 2. Okay. The chromosomes arrange themselves in the equatorial plane. Okay, equatorial plane with the spindle fibers attached to them. Start moving to the opposite pole. They are arranged in the equatorial plane, but now they will get ready to be pulled apart to the opposite pole, and that stage is known as the anaphase. So let's understand. Homologous chromosome was getting separated out, but the sister chromatic till now have not yet separated. So during this anaphase 2, the sister chromatic will separate. If you don't understand what is sister chromatic, I have told you many times. Suppose this is a chromosome, this is another chromosome. These two are known as the sister chromatid, and these two are known as non-sister chromatid. So each chromosome, by this you can see the definition, each chromosome consists of sister chromatid. Okay, this chromosome, these two are sister chromatid, and if two different, this is one chromosome, suppose this two second chromosome, the chromatid of two different chromosomes is known as non-sister chromatid. So now the chromosome is in this stage. Now what will happen, as I have told you already, the homologous chromosome have already separated out. But now the turn is to separate out this sister chromatid. Okay. So during the anaphase 2, the sister chromatid, these are the main event which I am discussing. Sister chromatid will separate. Okay, will separate. Okay. The sister chromatid, what is non-sister chromatid? Now what will happen? They will start. Suppose this is a cell, this is the two daughter cells. Okay, the sister chromatid have separated out, so the sister chromatid start moving towards the opposite pole. Okay, but still attached to the spindle fiber. The spindle fiber have not yet separated out. Remember this point. The spindle fiber is still attached. During the telophase, it will disintegrate. So remember the events which are occurring, because this cell cycle is very easy topic. Now main thing is to remember that during which event what is occurring. So still attached, and now you can see the sister chromatid have, before it was attached, okay, like this. Now they will separate out and move towards the opposite poles, and this phase is known as the anaphase. Okay, lost will be regained back. Okay, now they have moved to the opposite pole. Now what will happen during this telophase 2? These two daughter cells will start separating out. Okay, and they will divide from the middle. Okay, this will divide from the middle, this will divide from the middle. As a result, this will form two daughter cells, this will form two daughter cells. So total 4 haploid daughter cells are formed. Okay, total 4 haploid. As that I have told you how the cytokinesis process have occurred. So in the same way, the cytokinesis, what will happen? The nuclear membrane, this will all again regain back, okay, around the cell, and the spindle fiber will degenerate, as I have already told you. And now what will happen? The cytokinesis process, that is the separation out of the cell from the cytoplasm, it will divide into two. This will divide into two. As a result, by the end of telophase, we get 4 daughter cells which are haploid in nature. So by this we concluded that during the end of meiosis 1 we got 2 daughter cells, and by the end of the telophase 2 that we got the 4 daughter cells. But if you remember, in mitosis we were getting 2 daughter cells. Okay, that was equational. But here, why it is called reductional? Because the one cell is getting reduced to into 4 daughter cells. That's why it is called reductional daughter cell. Okay, reductional division. Meiosis 2 is called as the reductional division. Now we will understand what is the significance of this meiosis. Helps to restore the chromosome number. First part, 2n sperm, 2n. Then you know that the gametes undergoes the meiosis process, mitosis also, meiosis also. But by the end of meiosis, this will donate 1n, this will donate plus 1n. By the end of the zygote, will have 2n. That is, by the process of meiosis only this gamete formation occurs, if you know that. So this helps to restore the gamete, they restore the chromosome number. Okay, 2n means chromosome, that is diploid number will be restored back. Second, what will happen? Second significance is that it's a new opportunities for the combination with the cell, that the egg will have the opportunity to combine with the sperm. So as a result of which we will get one offspring. So they help in the opportunity for new combination of cell. And what was the significance of mitosis? Mitosis occurs in the, that is in the somatic cell, whereas meiosis occurs in the germ cells. So this is the difference between mitosis and meiosis. Now we will discuss a very important part, that is the calculation of the chromosome number and the DNA of this both mitosis as well as meiosis of each stage. Because in exam it will be asked that during the prophase how many chromosomes are present or what is the DNA count, and during mitosis that is what is the chromosome number and DNA count. A simple trick we will understand it, and how you can remember, you can note down this also chart that we will discuss about the meiosis 1, what is the chromosome number at each stage and what will be the DNA count. If you begin, if you know the simple thing that we have 46 number of chromosomes, that is 23 pairs, okay, 23 pairs of chromosome and 46 total chromosomes. So in the prophase 1, no reduction, no division have occurred yet. So the chromosome number will also remain the same. So that is 46. And if you want to know the total amount of DNA count will remain 92. Okay, 92 is the DNA count, because now the DNA duplication have not yet occurred. Okay, the DNA count is 92. If you double it, that is the DNA count. Okay, in the metaphase 1 also no division of cells have occurred yet. So 46. The DNA count will also be 92. Anaphase also will. Anaphase is also 46, 92. Why? Because here homologous chromosome have separated. The chromatids have not yet separated. So 46, 92. By the end of telophase, or you can say the telophase 1. Okay, telophase also 46, 92. By the end of telophase, after the telophase will over, you know that 2 daughter cells will be formed. That is, the 46 will be reduced to 23. 46 will be also reduced to 46. So by the end of telophase 1, you know that 2 daughter cells will be formed. Each daughter cell will be carrying 23 and 46. So this is known as the meiosis 1. Okay, reductional. How they have reduced the meiosis 1. With the 23, if you remember, by the end of telophase 1, we were left with 23 chromosome and 46 DNA. Okay. In the metaphase 2, same, no division have occurred. So 23 chromosome, 46 DNA. If you understand each and every step what have occurred, I have already discussed, then this chart is very easy. If you apply the trick that during the anaphase, or what was happening in anaphase 1, that is the homologous chromosome was separating out. Chromatid have not yet separated. So no reduction of cell. But by the end of telophase, the two daughter cells was formed. So they were reduced into half. This much is different. Rest of the things is same. Okay, 23. Remember, during the anaphase 2, what was happening? The chromosome number was divided, chromatid was separated out. So each chromatid will contain 46 and this will contain 46. Okay, this DNA count will remain same. Then during the telophase 2 also 46. But by the end of the telophase 2, this will reduce into 4 daughter cells. So each daughter cells will carry 23 and this daughter and the DNA count will also be 20. So by the end of telophase 2, each daughter cells will carry 23 like this. They will carry. Okay, each daughter number of chromosome from the very beginning, because this is mitosis. Then 92 number of DNA. Okay. Metaphase, what will happen? Same 46, 92 because no division. But during the anaphase, what will happen? The separation of sister chromatid will occur. Sister chromatid means separate, means it will become double. This will remain the same. This will also become same. Okay. Why? Because still double is going on. Now, by the end of the mitosis, it will reduce to 46 and this will reduce to 46. Okay. So remember, when the sister chromatid cells separating out, then they are getting doubled. Why? Because each sister chromatid will carry 2. Okay, 46, it will carry. That's why it is getting doubled, not reduced. But here, they are getting reduced. At the end of the, why it is getting reduced? Because 2 daughter cells are formed. As a cell is getting separated into 2 daughter cells. That's why they are getting reduced. But here, this chromosome will be same. Okay. But they are separating out into 2 chromatid. So each chromatid will contain 46. So total 92. Okay, remember that when the sister chromatids are getting separated out, then they are getting double. In the metaphase 2 also, during the meiosis 2 also, during the anaphase 2, that is the anaphase 2, the sister chromatids was getting separated. That's what it is getting doubled. If you see the previous chart. But during the meiosis 1, the homologous chromosome was getting separated, so there was no reduction. By the end only, they were reduced. So if you understand this 3 chart, if you pause the video, note down this and go through all this, the thing which we have discussed earlier, that all the point we have discussed, then you can understand how to calculate. Because this calculation part is very simple, very easy. If you understand what process are occurring in each step. Okay. So by this, we have come to the end of this meiosis 2, and in the next video we are going to discuss about the cell cycle checkpoints. That is, the cell cycle, each step there is checkpoint or the gatekeeper is present, obviously to check whether the process is going on smoothly or not. Okay. So by this, hope you liked it, and if you are new to my channel, do subscribe, like and share among your friends as much as you can, and good essential parts for your exam or interview part. So please do learn and have a nice day. Thank you.